My name is Antonis Rokas. I'm the director of the Evolutionary Studies Initiative here at Vanderbilt, and I would like to welcome you on this inaugural Biodiversity Lecture. We have many different named lectures that's evolutionary studies, such as Darwin Day, Earth Day, the Scopes Lecture, that I encourage you to explore or come to. But we realized with the seminar committee that we didn't have a series devoted specifically to life's diversity. And if you think about it, most of us, if not all of us, are in this room studying biology because we're really fascinated by the diversity of life. So this new biodiversity lecture series aims to fill that gap. And as the seminar committee was sort of thinking about potential names of, who would be that inaugural speaker or set of speakers, we really could not think of anybody better or more appropriate than our distinguished guest speakers today, Drs. Rosemary and Peter Grant from the from Princeton University. I think you all know that they've done some really groundbreaking research on Darwin's finches in the iconic Galapagos Islands. They've spanned several decades doing this research, and it's still ongoing. Rumor has it there'll be a slide from one of their latest papers that was just published this last Friday in the journal Science, and so they're not resting. And their work is literally in every textbook. Their work has deepened our understanding of how diversity is generated and unraveled the mechanisms by which species adapt and diversify in response to changes both in their biotic environments, how they interact with other organisms or occasionally hybridize, as well as abiotic environments, so changes in temperature, rainfall, and so on. I'm not going to list all the prizes they've gotten because they've gotten literally countless awards. Kyoto Prize, Darwin Wallace Medal. If you won that, you have to wait another 35 years because they bestow it every 50 years. And they got it in 2008. The Balzan Prize. They're members of the US National Academy of Sciences, members of the Royal Society of London, and so on. So Rosemary and Peter, we're really beyond delighted that you are accepting our invitation. So please join me in welcoming them to Vanderbilt.
Fine, thank you. Thank you for that lovely introduction, and of course, the invitation as well. It's been a great pleasure to come back here to Vanderbilt after a period of about 5 years or 6 years, when we had a wonderful visit beforehand. And I'm going to start, I'm going to give this first half of the talk, and then Rosemary will follow with the second half. And I'm going to start by saying that a major challenge for us evolutionary biologists is to explain the enormous biological diversity of this planet. There are millions and millions of species. And we are so ignorant, we don't know how many there are on this planet to the nearest million. That's a shocking statement, but on the other hand, it's a wonderful challenge for members of the audience to join the enterprise in trying to explain biological diversity. Well, consider, for example, a small group of, relatively small group of organisms within this massive diversity that we have, adaptive radiations. That is, groups of closely related organisms that are derived from a common ancestor relatively recently and relatively rapidly. So, for example, the cichlid fish of the Great Lakes of Africa are probably the most famous because hundreds of species have evolved, according to what I just said, relatively rapidly from a common ancestor in several of the different African lakes and also some in South America. That is probably the most spectacular example of adaptive radiation that we know, but there are many others as well. And here I'm showing a picture of anolis lizards in the Caribbean. There are 135 species, all derived once again from one or a very small number of ancestral species. And thirdly, finches on the Hawaiian archipelago. Honey creeper finches, they're most related to finches. Now, there are 50 species that we know about having evolved. Unfortunately, half of them have become extinct, in part due to anthropogenic activities. For groups of organisms like any of these, I could have picked Drosophila as well, also from Hawaii. Here are the 4 kinds of pieces of information we want, and before we can say that we understand the evolution of that group. We need to know the history of the group as best as we can. We need to know the environment under which the radiation took place. We need to know the traits that have been very important in the radiation, and we need to know the genetic basis of the variation in those traits. This is a big undertaking, and it demands research attention across a very broad range of scientific disciplines, from molecular biology on the one hand, all the way through to biogeography and paleontology on the other. Now, in this lecture, we will tell you what we have been able to learn from a study of just one group of organisms, Darwin's finches in the Galapagos Islands, carried out over more than 40 years ago. There are no fossils of the group, so from genetic data, we've been able to reconstruct inferentially the pattern of evolution of the group. And we know from mitochondrial DNA that these 18 species that I'm showing on the phylogram evolved in the relatively recent time of just 1 million years, 18 species in 1 million years. They've diversified mainly in body size and beak shape, not very much in plumage, not very much in color or in pattern. Now, beaks are tools for exploiting the environment, picking up food. And so this is a radiation that is primarily ecological, fostered by the relative absence of other species of competitors and predators due to the remoteness of the islands. The islands themselves have had a very dynamic history. They're volcanic. They lie on a plate called the Nazca Plate, and they are moving towards the South American mainland, slightly southeast, at the rate of about 60 to 80 kilometers per million years. They owe their origin principally to a hotspot south of Fernandina, currently south of Fernandina here. That plate is moving then this way, carrying the islands that way. And so geophysically, this is a dynamic environment. Over that same 1 million years, that last 1 million years, there have been major upheavals in our climate, the global climate, that has affected Galapagos. There have been 10 full glacial to interglacial cycles of oscillations in temperature with a very strong influence on sea level. So sea level, sea levels, even in Galapagos, remote from the polar regions where ice is melting or building up, the sea level has changed. So I'm illustrating with 3 pictures in what it was like over the million years during the course of one glacial interglacial cycle. So the water went down at a time when water was locked up in ice in the polar regions, and so islands became higher and bigger and closer together, and even coalesced into even larger land masses at the lowest level of the sea level. And I'm not going to do this, but I could reverse these pictures to show what it was like in switching from a glacial part of the cycle to the interglacial. We're in an interglacial now, so that the islands don't look anything like this, but they did. They do look like the pattern that you saw in the first picture. So the islands, against this dynamic background, how do we explain the evolution of this group of organisms? Well, key to the answer is identifying the causes of speciation. That is the divergence from a common ancestor into 2 lineages that ultimately become species. And so I'm illustrating this with a simple Y diagram, where ultimately the products of this divergent evolution are considered to be species because either they don't interbreed, or if they do interbreed, they interbreed with relatively little success. Now, our ideas about this process go back to Darwin, who identified spatial separation of the lineages as a key determinant of divergent evolution, starting the process of leading up to full species formation. And I'm illustrating his ideas with a very simple diagram. Everything is hypothetical here, and it involves three things. One is dispersal of organisms from the mainland to the islands in the first place, and then further dispersal around the archipelago, with change taking place at the various steps. And the reason for that is, first of all, random genetic drift, the ever-present random genetic drift will cause some change in the populations derived from another one. And then adaptation to resources in each of the island environments, because the resources are not identical. And by resources, I'm talking about food principally and nest sites, because these are birds. And then finally, there is likely to be further divergent evolution where two lineages come together, arbitrarily chosen to be on San Cristobal here in the southeast. And there, natural selection is to be anticipated, causing further divergence and minimizing two things: interbreeding being one, on the assumption that the products of interbreeding are going to be relatively unfit. And the second one is competition for the resources that I mentioned earlier, food and nest sites. And I'm going to give you an example of that, called character displacement, from the little island of Daphne, unmarked, north of Santa Cruz Island, where we've been studying a microcosm of the adaptive radiation for getting on for close to 50 years now. This is the island. It's called Daphne Major, and it is 120 meters high, 3/4 of a kilometer long. It's never been settled by humans. There's nothing there for humans to do. And so it's in a pristine state. It's occupied now by 4 species of finches in the single genus Geospiza, or ground finches, ground finches. So there's a large, a medium, and a small version, and then a relatively long beak finch, with a cactus finch. I'm going to interrupt the scientific part of this talk just to give you a little idea what it's like to live on the island, because we're often asked this question, and we've got now into the habit of telling people that we put into quarantine all of our food and supplies for 48 hours before we get onto the island to minimize the chances that we're transmitting to the island things that shouldn't be there, from microbes onwards. In the center of the island is a crater. The birds nest and fly everywhere. They nest almost everywhere. We go almost everywhere following the birds, and to establish their breeding. We live in a tent, or rather we sleep in a tent. We spend much more time in a cave where we cook. And this little cave, open to the air, doubles up as a laboratory. Like other ornithologists elsewhere, we put up mist nets to capture the birds. They fly in, they don't see the nets, they get tangled in. We disentangle them. They're not harmed by this at all. We take them out and we take them to shade. And this time we take to another cave where shade only lasts until 10 o'clock in the morning. And here we process the birds. We weigh them. We take measurements, linear measurements, beak and body measurements. And I put on the legs of the birds 4 bands, 1 metal band with a number punched into it, a unique number, and 3 plastic colored bands, color-coded to correspond to the number on the metal band. And then when we release the bird, it's carrying an individual identification, identity. And So we don't need to capture it again in order to identify a bird so banded, doing various things of interest to us in the environment. But before we release the bird, Rosemary takes a small drop of blood from the wing vein, a little prick there with EDTA covering it, a piece of filter paper put across there, then with EDTA preserved, put into a jar after it's dried, into a jar of dryerite, and then transported back to the United States for extraction of DNA and analysis thereafter. Then we release the birds and they fly away unharmed. So back to the birds. I'm showing here a morphological plot of all population averages across the whole of the archipelago of the 4 species in question. And on Daphne, the principal ones are Fortis and Scandens, with population sizes of about 100 breeding pairs and 50 breeding pairs, respectively. The first of these, Fortis, the medium ground finch, is a granivore, seed eater, a generalist. Now, from all of the work that we've done to band the birds with unique identifiers on their legs, we have been able to watch literally hundreds feeding and compile an inventory of their diet. And from that information, we know that those birds seen feeding only on small seeds have small beaks, and those feeding on large seeds, as well as small and medium-sized seeds, have very large beaks. So the species is a generalist, but there's some degree of beak-related specialization. The other principal species is the cactus finch, Scandens, and here all members, it is the species is specialist and all members do the same thing. So they probe into open cactus buds and then probe into the flowers for nectar and pollen. They with this hammer-like, with their chisel-like beaks rather, they hammer a hole in the hard cover of the fruits, extract seeds, and then with their strong beaks, crunch them open to get at the kernels on the inside. Now, the reason for thinking that they may be evolving now is that they're at the tips of the phylogenetic tree in this blue zone here. And not only that, but they have been evolving rapidly at that tip. And there has been, in just recent times, a rapid accumulation of species. So there's not a slowing down of the production of new species, but rather the opposite, an accumulation of new species formed. So, are indeed, is evolution in fact happening? And if so, what does it tell us about speciation? Well, the birds are certainly evolving, I can say that. Here's a plot of the average beak depth of the medium ground finch, Fortis, over a 40-year period. And during this period, there have been 3 major perturbations of an otherwise stable currents of beak size across years. Here in the 1970s, there's one major increase, a smaller decrease in the 80s, and then in 2004, a very large decrease. Natural selection was involved in each case, and so I'm going to describe each one separately. But there's a common denominator, and each involved a drought converting vegetation like this into vegetation like that. 0 further production of small seeds, and for the finches, mass starvation and mass mortality. And so the first one involved a drought in 1977, where as much as 80% of the Fortis population died. And at the beginning, all finches fed on the small and easy to handle small seeds. But then as they depleted their supply of small and easy to handle seeds, the finches had to turn increasingly to large and hard seeds, including this one that were still present and in increasingly relative abundance. Like this one, tribulus, which requires quite a mechanical force to crack open to get to the seeds on the inside. And so as a result, the average beak size started to increase from that point onwards and didn't cease until the rains came in early 1978, alleviating the stress on the population and allowing the survivors to breed. So natural selection had occurred from the beginning before the drought to that period of 1978. Now, for evolution to occur, which is a change in genetic composition of the population from one generation to the next, the trait in question, beak size in our case, has to be heritable. And indeed it is, as shown by these two plots that plot the average of the daughters and sons' beak sizes against the average of the mother and the father. In short, the offspring beak sizes are highly predictable from the parental beak sizes. The heritabilities, which is a measure of how strong that is, are shown here, which are the slopes of that relationship. So, not surprisingly then, in the next generation, evolution occurred through a combination of strong natural selection on a trait that is highly heritable. And there is the plot that shows the evolutionary response, and the predicted one is there. So the offspring produced, the survivors produced offspring that were very similar to themselves as a result of inheriting genes that affect beak size in the same way that they did affected their own parents. And I'm going to summarize by saying then that during this course of a drought and following, natural evolution occurred by natural selection on a heritably varying trait for mechanical reasons that we could understand when the environment changed. The next episode involved another change in the environment, but first I'm going to have to tell you about El Nino, because this is what started it all. An extraordinary El Nino event occurred in 1983 with an unprecedented rise in temperature and amount of rainfall. According to Coral Core data, this was the most intense El Nino event of the last 400 years. It certainly was an enormous amount. As you can see from Daphne, more than a meter of rain fell on this semi-desert island. But notice that 2 years later, there was virtually no rain. So another serious drought, but under different circumstances, which I will illustrate with these pictures. This is what the island would look like under normal dry season conditions. Note the cactus bushes, because they're going to be markers for what I'm going to show. And so the rains come, and then everything turns green on the island. The plant that made a difference between survival and non-survival in 1977 is this one, tribulus, growing low over the ground. And the rains came so heavily that the other plants responded so strongly as to smother this plant, as you will see from the next picture. Just rampant growth everywhere, so much so that the cactus bushes were almost completely smothered by vines. Grasses and vines alter the environment and change the composition of seeds from an emphasis, as it were, on the large and hard seeds of tribulus to an absolute superabundance of small, soft seeds. One could recognize the effects of El Nino even in the following 1 and 2 years. And during the 1 and 2 years, the rains were either very weak, 1984, or absent altogether. And under these circumstances, this pendulum of natural selection swung in the opposite direction from 1977. Now birds with small beaks, with small pointed beaks in particular, capable of picking up and processing large numbers of very small seeds to meet metabolic needs, had the survival advantage. So they increase in relative frequency. The large beak members started to die off at a higher rate, and the average beak size of the population shifted towards small size. The third event involved a different factor. In that El Nino event, a new species was added to the breeding list. 2 female and 3 male Magnorostris immigrants stayed to breed rather than flying into the island and then back to their island of origin. They stayed to breed and they bred successfully. And over the years, numbers built up. So by the time that the next really stressful occasion occurred on the island in 2004, a serious drought in 2004, there were about 150 of these individuals on the island. Now you can see by looking at this big beak here, what powerful force could be applied to the tribulus fruit on the inside. And you're probably going to guess that these birds were super efficient at feeding on tribulus, more so than Fortis, the medium ground finch, and they outcompeted them. So this one, the Magnorostris, outcompeted the large members of the Fortis population for the diminishing supply of tribulus, as shown here. And so they went out, not completely, but relatively their numbers went down. Not completely, still some survived, but the survival was greater amongst the small-beaked birds. Now, they too were suffering, but they were suffering at a lower rate than the large-beaked birds. And so they had the relative advantage, and the average beak size of the population went plunging down to this point here. And the net result was an increase in the difference between the immigrant Magnorostris that had come in and the resident Fortis that suffered the competitive influence of Magnorostris. And that's an example of character displacement, which is the divergence of populations that were previously separated and come together and compete for food, which I used to illustrate. Well, I mentioned this when I said, there's Daphne, where I will give you an example. That is the example that I was referring to. So we know quite a bit then about the phenotypic variation of the population of Fortis and the relationship to fitness under ecological circumstances. But now what about genotypic variation? How does that map onto the phenotypic variation? Because after all, the genotype is part of the whole story. Well, to investigate in detail genes underlying the important trait of beak size, we collaborated initially with a developmental genetics group at Harvard University, spearheaded by Cliff Tabin. Archa Abzhanov was the doctoral student at the time who did the work, most of the work, and later Ricardo Majorino, who is now on our faculty. So between, first of all, Cliff and Archad managed to establish the first of 3 developmental pathways affecting beak size differences between species. So they found by their analyses that BMP4, bone morphogenetic protein 4, a signaling molecule, was graded in its expression from the deep-beaked Magnorostris species all the way through a linear gradient to the one with a shallow depth of beak, namely Scandens. Now, this was just one of 3 signaling pathways independently affecting the 3 dimensions of depth, width, and length of the beak. There's BMP4. Calmodulin is a separate pathway affecting beak length, and beak width is influenced by another signaling pathway later in development when cartilage is being condensed into bone. So you can see that tweaking any one of these three pathways would alter beak shape. And that could account for at least part of the reason why species differ in beak shape. To find out now the genetic control over variation within species regulating beak development, we turned attention to a collaborative arrangement with a different group of people. Leif Andersson is a genomic specialist in the University of Uppsala in Sweden, and there's his group. We've collaborated with him for a decade, and here the question has been, can we identify the regulators of variation in beak development within our fortis and within our scandins and so on? And they have been able to discover many things of great interest. The two principal initial genes that they discovered, both transcription factors, were HMGA2 and ALX1. ALX1 variants affect beak shape variation. HMGA2 variants affect beak size variation. And there is our first principal result. Now, you're probably, many of you, if not all, I don't think all, will have seen plots like this before. The way the research goes is to sequence the whole, in our case, the whole genome of all of the finches, and then put them into phenotypic classes that are contrasted, and to look for the differences in the genome between the phenotypic classes that might then be candidates for explaining the differences in phenotype. The actual pictures there are shown horizontally between, now I'm going to pinpoint, large versus medium, medium versus small, and the top one as well. But I think of them as vertical strings of chromosomes all linked up together, where you run down the two strings, large versus small, and you ask phenotypically they're different, but where are they genetically different? And then you find one region where the strings don't match. And in our case, we found it one region here, 512 kilobases in length, where there were 4 genes, one of which, HMG2, is known to be associated with body size variation in several mammal species, dogs and rats and cats and mice, and even us humans as well. Well, in Darwin's finches, variants at the HMG2 locus actually correlate more strongly with beak size than with body size, very strongly, in fact. So one is associated with large size, and therefore we give it the LL for large symbol, another for small size, and then the intermediates are, as expected, in intermediate size. And not only that, but during the drought of character displacement that I mentioned, when small beak birds had an advantage, phenotypes of a small size had the advantage. The genotypes also showed a very strong differential survival. So the LL genotype had very low survival. The SS genotype had very high survival, and once again, the intermediates had the intermediate survival. The selection coefficient against large genotype was a very high 0.59. This is really truly remarkable. This was the strongest change in allele frequency across the whole of the period that we have analyzed the data. And to bring you right up to date, we have, with our collaborators, identified altogether 28 genes of association with beak size or beak shape, of which, in the most recent investigation, 6 of them have been identified as independently associated with beak size development. And 2 of them, 07 and 03 up here, have a remarkably high explanatory power of about 50%. 50% of the variance in beak size or beak shape can be explained by these 2. Now, that is an enormous amount for just 1 or 2 single genes. So I'm going to summarize at this point and just say that I've talked about natural selection and and described how it occurs and when it occurs, for the reasons why it does occur. I mentioned that other species determine the direction of the evolutionary response to natural selection. So the first one you can think of as being associated with allopatric stage of speciation, and the second one, the sympatric stage of sympatric speciation. The sympatric stage of speciation is associated with that second example of a character displacement. So both of these factors are the core ingredients of speciation and treated as an ecological phenomenon. But that leaves unanswered the question of reproduction. So how do the species actually keep apart? And for that, I'm going to hand over to Rosemary. Yes, that's right. That's key.
Okay, so Peter has told you all about these now 4 species that occur on Daphne. This tiny little island is sometimes, particularly after an El Nino event, full of birds. So we have tortoise, which when they're breeding can have almost 1,000 individuals on this island, and they drop down to less than 100 during droughts. Then there is scandens, which goes up to about 750 individuals, dropping down to below 100 individuals. Magnerostras that came in that have built up a population of over 200, and then very occasionally Polygynosa comes into the island as well. So they are all members of the same genera. So how on earth do these species keep apart? What are the reproductive barriers? So I'm going to tell you about the causes and evolutionary consequences of rare episodic gene flow between the species. Most of the time they're kept apart. So first of all, I'll deal with why they're kept apart. And then I'm going to talk about the formation of a new lineage, which we have followed from its inception right up through 6 generations. So what are the reproductive barriers? And somebody's going to ask me this question: what do I consider a definition of a species? So we take the biological species concept, which is species are reproductively cohesive group of organisms that are diagnostically distinct, and reproductive barriers between them are rarely penetrated, very rarely penetrated. So they cannot use plumage because most of the males, or all the male ground finches, are black. Females are brown or grayish brown, and also they have the same courtship displays, and they build the same type of nest. But where they do differ is they differ in size, from a 32-gram Magnerostris to a 12-gram Polygynosa, and they differ in beak shape, and they also differ in song. And song, it turns out, is very important. And so we can tell the birds apart like this, but actually, first of all, we had to make sure that the birds could also discriminate between individuals of their own species and individuals in other species. So to test whether they could do this by morphology or just appearance, we took the Stuff Museum specimens of female porters in one case and female scandens in another, took them at the height of the breeding season into a territory and asked, could the living male discriminate between a female of his own species and a female of another species, even though this female was just a Stuff Museum specimen? And the answer was a resounding yes. They vigorously portrayed a female of their own species and completely ignored the others. And we did this with different combinations of species. We did it with controls, and the answer was very definitely that they could discriminate between their own and another species purely on the basis of morphology in the absence of song. So could they discriminate by song in the absence of any morphological cues? So to do this, we recorded song, played back song. When we played back Porter's song, Porter's came into the loudspeaker, scandens went on feeding or doing whatever it was doing, and ignoring it, played back scandens. song, and then a birth happened. Scanders came in and Fortis ignored it. So one thing we did know about song is we learned this from Bowman, who did experiments in the early 50s, 1950s, where he took birds into captivity, which you couldn't do now, and he showed that song was learned early in life during a very short sensitive period of 10 to 40 days after hatching. Now, all this time, the male is singing. Females don't sing, but the females, both males and females, were feeding the offspring, and it was the male that was singing. So they learnt their father's song, their father's species song. And we know that this song is retained for life. We record the birds every year, and we have long strings of song. So we know it's retained for life, but we also know that as adults, they pair according to the song they learned early in life. And we have never seen an exception to this, not just on Daphne, but in all the other islands. They always pair according to the song that they learned early in life. Now, this pre-mating barrier is vulnerable because it's based on learning, and it can be breached if a young bird hears and learns the song of another species during its sensitive period. And we have about 1% each breeding season of a bird doing this. And it usually does this when two things happen. Either it can be a nest takeover and an egg is left behind. This is really very rare. More often it's due to the death of the father. Females don't sing, and the birds hear a bird in the neighboring territory, and if that neighboring territory is a bird of another species, they learn that species song. So this happened when Magnorostros colonized the island in 1983, and over the next few years, we had 8 Fortis and 2 Scandens that learned and sang a Magnorostros song. So this is a Magnorostros song. This is a normal Fortis song, and this is one of the Fortis singing a Magnorostros song. A normal Scandin song, and a Scandin singing a Magnorostros song. Now, none bred with the Magnorostros, and the reason was that when the next breeding season, when these little birds started to sing, so you can imagine a little bird starts to sing a Magnorostros song, a Magnorostros almost twice the size of it just rushes in and just beats the living daylight out of it. So nothing happened. The birds certainly didn't breed, and the birds didn't survive. So it seems that when the size difference is very large, this barrier is robust. Even learning another species song didn't lead to hybridization. However, when the size difference between species is small, like the 17-gram Fortis to a 22 roughly gram Scandens, learning another species song can lead to hybridization. And we were very excited about this because we had followed all these birds and we had a few, very few hybrids. And we thought, gosh, could we see some beginnings of genetic incompatibility between Fortis and Scandens? But for the first 10 years of the study, we followed these hybrids and none of them survived the drought season in order to breed. So we thought, aha, maybe there really is some genetic incompatibility that we'll be able to ferret out. But maybe there's nothing there because many of the birds were dying anywhere, and it may not be, they may not have had enough food. And this actually what was going on. So during the El Nino, vast amounts of small salt seeds were produced. The birds, the hybrids survived, and they bred according to, they followed the rule, they bred according to the song that they learned early in life. But we could still ask, how well did they survive? So what we did is we took the next 3 cohorts when there was a vast amount of food in the environment, and this is the survival of Fortis and Scandens in these 3 cohorts when there's a large amount of food. And we asked how well did the hybrids survive compared to the parental species. And the answer is that the hybrids survive as well as, if not better, than the parental species. So we had no evidence of genetic incompatibility, and the hybrids, as I said, back crossed according to the songs they learned early in life. So at this stage, all we had from our blood samples was microsatellites. But even with microsatellites, we were able to see that Porter's genes were passed into scandins, and that Porter's and scandins genetically began to converge. And this followed the pattern of morphological pattern of our measurements, which also showed that Porter's and scandins were converging. So we were excited about this, but after all, they were only microsatellites, which in those days was really something quite big, but now it seems like absolutely nothing. But the other thing that we were noticed, that integration was mainly, not solely, we had a little bit going the other way, but mainly from female porters into male scandins. And the reason for this was that scandins' sex ratio was highly skewed at the time, and basically still is, in favor of males. So the males were desperate for females, and they mated with the hybrid females, whereas the hybrid males were outcompeted by the larger scandins for both females and territories. So, but this was able to show that this is what actually another pattern of what happened was that this is where introgression began about here, and over the years, even though it was just a trickle of genes, scandins were coming, the beaks were coming blunter and blunter and blunter and more tortoise-like. So we wanted to know if there were specific alleles that could have contributed to the changes in beak shape. And once again, we collaborated with Leif, who is here, and the person, his graduate student this time was Sangeet Lakamani, who came from Nepal, and he did most of the work. So the first thing that they did was exactly, I won't go over it in great detail anymore, but it's exactly what Peter said. They took all the blunt beak birds and all the pointed beak birds from all our blood samples from all islands we visited, and they found that what popped out was this transcription factor, this gene, which is actually a transcription factor, which we were very excited about because ALX1 also occurs in humans, and it's very well known, particularly amongst dentists, because a mutation in ALX1 causes cleft palate in humans. Now, in the birds, it comes in two forms. All the ones with sharp pointed beaks have one form of ALX1. In blue, all the ones that are blunt, or the Magnolostrus and Conilostrus on Española, have very blunt beaks. They were homozygous for ALX1B or blunt. And Porters had both, mostly blunt, but they also had both. So one of the things we did was to make sure that they really were acting in the right place, as we suspected. And so we turned to Arka Abshnok, who had done the previous work that Peter showed. He was now full professor in London, and he was able to show that yes, indeed, ALX1 was expressed in the developing beak in all Darwin species. So, so as I said, that this is just really to repeat that it goes from female hybrids back into Scandinavian. And so they were, Sangeet and Leif, other people in Leif's lab were able to track exactly how the ALX1 blunt allele moved from Portus into scandins to produce the scandins, which now contain the blunt allele. and it became much blunter. And you can even see this, all these birds were caught in a net. The same net in 2012. This is Scandins with much blunter beak and had the B allele, ALX1B allele. This also had the ALX1B allele, but this one did not, and is exactly the same as Scandins before introgression occurred. So it has a long pointed beak with the ALX1 pointed, homozygous for the ALX1 pointed beak. So Peter and I wrote in one of our papers that a mosaic genome arising from hybridization between two species generates phenotypic novelties that would be under selection in a new or a changed environment. And because the increased genetic and phenotypic variation could fuel a rapid change along a different trajectory. So that was all made sense from what we knew, but we had never seen it until this time. And so we were absolutely bowled over to find that we could follow this in the population of finches on Daphne. And this was in the formation of a new lineage of finches. So what happened was that a finch arrived on the island at a time when all the finches were banded. Trevor Price, who some of you may have heard of, was our graduate student at that time. And so he caught this bird, he weighed it, it was 28.5 grams, whereas a normal tortoise is 17 grams. So it looked like a giant tortoise. And it was a young bird when it arrived. It very quickly, much quicker than tortoise, got into fully black plumage. And the next year it started to sing, and it had a completely unique song, never heard on Daphne before. We came along and we caught the bird again, and we took a blood sample this time. So we had a blood sample from it. And what it did is, well, first of all, I say, where did this bird come from? Peter and I initially thought that it must have been a hybrid from Santa Cruz, the nearest island, but we have both written in our notebooks, it looks awfully like a Conirostris from Hispaniola, but we thought this is impossible because Hispaniola is away in the south of the archipelago and it couldn't possibly have flown from Hispaniola. But Sangeet in Leif's lab did all the genetics and found, yes, indeed, it was a Conirostris from Hispaniola, who somehow, as a young bird, had managed to get to Daphne. Now, it was a brutish bird. It was a big bird. It managed to get the best territory on the island, lots of cactus bushes, and it sang lustily, but it was all the wrong size, or the wrong shape, or the wrong song, and it couldn't get a mate. And it sang for 2 years, and without getting a mate, even though conditions were perfect, until the very end of the second year, the last breeding season, a female tortoise had lost her mate. The male had died, and she moved into this wonderful territory, and she bred with him. And then another female tortoise did the same. So I'll go back. So we followed all these birds and followed took blood samples from all the offspring, and we found that all of them were truly had genetic transmission from 5110, so there had been no cockabee or anything like that, and all males sang 511 and song. So it followed the rule that all the males sang their father's song. Now, then along came the 2.5-year drought, which caused the character displacement event that Peter told you about. 90% of the birds on the island died. All these birds died, except for these two, a brother and a sister, an inbred brother and sister. So 2.5 years later, when the rains returned, the inbred brother and sister bred with each other. They produced 26 offspring, all but 9 survived. We had a daughter mating with her father, a son mating with his mother, and the rest of the brothers and sisters, the fibs mating with each other. To produce more offspring, and all of these birds we caught and followed, took blood samples from, and they all show genetic transmission from 5110. All males sang 5110 song, and all were large like 5110. So the inbreeding coefficient based on genome-wide heterozygosity went steadily upwards, as you could imagine. But what I think is the most exciting and most important was that there had been an allometric shift. So these, we now call them the big bird lineage. The big birds were not intermediate between porters in green and conidostris in red, but they were displaced. So they had a relatively large beak for their body size. Now you don't often see a shift in shape. You see much more often see a shift in size, but not often with a shift in shape like this. And the reason is that HMGA2, which Peter told you about, which was a body size transcription facilitator, and ALX1, which is a beak size, a beak shape, are very closely linked on the same chromosome, only 7 megabytes apart. But what is exciting about it is there's a recombination hotspot between them. So you can actually tweak one and get with this recombination between ALX1 and HMGA2. Remember, HMGA2 had we had the small and the large one, ALX1 pointed and blunt one, and you can get all combinations. So this is what was happening in the big bird one. And Sangeet was, well, we were all with all our data, were able to look to see if there had been any selection over the 6 generations that we had followed. And what happened is that the beak body size remained the same, practically flat, but there had been, although the sample size was very small. So in 2009 was really the only year when there was a sufficient sample size of 19 adults. Sorry, not 9, sorry, there were 19 survivors, but the 19 adult survivors had significantly deeper beaks than the 5 that died. It's significant, but you've got to remember that it's a very small sample size. But it's this recombination event between ALX1 and HMGA2 that probably shifted this. So these birds, the big birds, group were going around with Magnerostris, and they were eating tribulus seeds. So we thought, well, if there's another drought and the tribulus is left behind, they will be faring very well. So we were wondering how efficient they were compared with Magnerostris at cracking tribulus shells to get at the small seeds inside. And we found that by following them and measuring their cracking times, they were just as efficient as Magnerostris. Portus was now so small, it was basically ignoring the large hard tribulus seeds. But we argued that if there was another drought that was bad and tribulus was left behind, the big birds could even out compete Magnerostris, because they, being so much smaller in body size, they would be able to, but just as efficient in beak size, they would be able to survive on less food than Magnerostris. So the new lineage is behaving like a separate species. It's much larger than its nearest relative, Portus. On a morphological plot, it's Magnerostris here in green, Portus here, here. And then when Magnerostris outcompeted the large Portus, which was up here, it left a gap, and the big birds moved right in. It differs in song from all species in Daphne. This is 3 generations of the big bird song, very different from Portus song, Scandian song, and Magnerostris song. They hold contiguous territories on one part of the island. The blue dots are territories before the drought, the red dots after, and the more red dots over the cliff. These have large territories. They depended against each other, but the territories overlap with Portus Scandens and Magnarostras who they ignore and are ignored by them. So in all respects, the new lineage functions as separate species. Will it die out through inbreeding depression? Well, yes, it might. Will the genetic variation be augmented through gene exchange? This we think might be quite likely. We were on the island just before COVID, and we did see big birds then, but we don't really know what's happening. But whether it survives or not, this new lineage gives insight into how a new species could arise and either persist or become extinct. So in summary, we started in 1973, long before practically everyone in this room was born. And but there were only two species on the island, Portus and Scandens. After the El Nino year of 1983, they began to, integration began, and they began to fuse, happened down here. Magnarostras came onto the island at that time, outcompeted the large Portus, which was up here, and the big birds moved right in. So this given insight into three models of speciation: the allopatric model, which was the Darwin model that Peter showed you, fusion between Portus and Scandens. Scandens now looks very different from Scandens on any other island in the archipelago, and then integration, where we got with the big birds, where we got what is officially called homoploid because the number of chromosomes don't change, hybrid speciation. Now, how important is integration as a source of variation for adaptation to new environments? When we first started to publish this, I think our first paper was turned around with birds don't hybridize. So that was a long time ago. Now we know that hybridization and integration is everywhere. Well, we knew this before about bacteria. Horizontal gene transfer is very important. In plants, Lauren Wiesberg's shown that this homoploid hybrid speciation is extremely important in sunflowers, and Richard Abbott in Senecio. And in animals, it happens in every single animal. You see this, including ourselves, where there is hybridization and integration between the Denisovans and Neanderthals and modern human lineage, as has been shown by Svante Paavo and his group. And this has very much been a source of variation used in adaptation to new environments, such as adaptation to altitude, but also just more recently to COVID, where there has been a gene that is associated with a genetic risk for severe COVID was inherited from Neanderthals, but so was a genetic region association with protection against severe COVID-19. So this gives you some ideas that we have Neanderthal and Denisovan genes in us. So we many postdoctoral graduates, postdoctoral fellows, collaborators, and our daughters who are with us for long periods of time, the Galapagos National Park, the Charles Darwin Research Station, and our funding. None of this work would have been done without them. I just want to leave you with two messages. One, that you live in exciting times. The genetic data are rapidly accumulating. This enhances our field studies, but reciprocally, and I really want to stress this, a reliable interpretation of the genetic data requires a true understanding of ecology, evolution, and behavior in nature. And often this is forgotten by funding agents who would rather just fund genetic data. But in the theme of this, I love this NASA image because I think it captures the uniqueness, and it also captures the fragility of our planet, with biodiversity being its fundamental feature. And we've shown you in this talk that environments and populations are dynamic, they're constantly changing, and for a healthy environment, we must keep them both capable of further natural change. Thank you.
Great. If it's okay, we can go ahead and have a few questions for you, Joel.